Okay, we'll complete this short series of lectures with some discussion of exogen metabolism, particularly the reference to energy. So what we're going to look at, the main focus in terms of energy is going to be alcohol metabolism, but we need to get some rubbish out of the way first. This is all tosh, utter tosh. So-called detox diets have no scientific basis of the claims they make. The liver, as our main detoxification organ, has been doing a job splendidly for probably billions of years in our evolutionary history. Similar processes are found in fishes, for example, who diverged from us a long while ago. Okay, so some definitions. Drug is an exogenous substance that affects physiology and psychology. Now, exogenous means originating or produced from outside the organism. Also refers to substances present in high concentrations and are usually found in the body. Conversely, we have endogens, substances which originate and produced by the body are part of normal metabolic processes, part of its normal biochemistry. Exogens you'll sometimes see referred to as xenobiotics. So metabolism of exogens. Typically when we ingest exogens, such as drugs, our biochemistry tries to get rid of them, usually by excretion of the urine. Most of this chemistry takes place in the liver, which is often involved in increasing the polarity of the molecules, so therefore making them more water soluble. Polarity, as you'll probably remember from biochemistry, is a charge distribution of a molecule. The reactions that do occur here involve redox processes and also linked to energy metabolism, which you will discuss later in other modules. They take place in something called the hepatic microsomal enzyme system. Hepatic just means it's the liver. They take place, so for example, when we discuss alcohol oxidation in a short while, these reactions take place in the smooth endoplasmic reticulum of mitochondria in liver cells. Cell biology lectures will discuss these in more detail, these structures. There are some definitions there in the notes if you want to have a read on them. Again, as we see often in biochemistry, it's a structure with a very big surface area, so allows a lot of reactions to take place. As mentioned earlier, our biochemistry tried to get rid of exogens like alcohol by making the original molecule more polar. It does this by two types of reactions. The second class conjugations involve the metabolism of molecules like paracetamol. For alcohol, the chemical process involves just changing a functional group, so it's said to be a type 1 functionalization. So alcohol metabolism is a case study. What's the most significant difference between alcohol and other drugs we might consume? And maybe pause the video and have a think about that for a moment. This is the most significant difference. We consume gram quantities of alcohol, those of us who drink alcohol. Pharmaceutical drugs, milligrams at most, often micrograms. So a big difference in quantity. Some general physiological considerations. Consumed alcohol rapidly spreads across tissues and can't be stored. Like other molecules we get energy from, for example, lipids, carbohydrates or proteins. Most alcohol is metabolized in the liver, which is why this organ is subject to damage in the case of extended or excessive consumption. We do lose some by other means, such as sweat in the breath, and which is why breathalyzers work, and in the urine as well. But we can't store it, we either metabolize it or excrete it. There are three main routes, of which the ADH, alcohol dehydrogenase, is the most important one. It's found in everyone. The chronic drinkers tend to have enhanced use of the MEOS and catalase results. These are not discussed here but can be found in most biochemistry textbooks. All three routes convert ethanol first into acetaldehyde and then into acetate, acetic acid. Although ethanol is a polar molecule, acetaldehyde is more polar than it, and acetate is even more polar, so facilitating excretion, in principle anyway. The ADH route seems to evolve to deal with alcohol produced by anaerobic bacterial fermentation in the gut. This alcohol goes straight to the liver via the portal vein and has little in the way of physiological effects. We note that the ADH enzyme is also involved in the oxidation of many alcohols, steroids and fatty acids. It's a redox process of methanol to acetaldehyde to acetate. it involves alcohol dehydrogenases and aldehyde dehydrogenase. The dehydrogenase is a clue to their function. We'll notice that also at each step of the reaction a molecule of NAD is reduced to NADH. Pause the video if you want for a moment to think about the other side of the redox process with respect to ethanol. Okay, moving on. Just to summarise, the number one carbon of ethanol is oxidised acetaldehyde, noted as lost in hydrogen and gained in oxygen. Similarly, the carbon in acetaldehyde loses hydrogen and gains an OH where it's converted to acetate. In both cases these are oxidations. At the same time, a molecule of NAD for both processes is reduced to NADH. If you haven't heard of NAD, I'm sure it will be covered in cell biology at some point, and we'll explain what it is in a moment. The point about this is it uses lots of NAD up. Each molecule of ethanol uses up 2 molecules of NAD in its conversion to acetate. So this is back of envelope stuff. Vodka is about 40% alcohol by volume. Ethanol has a density of 0.78 grams per mil and its molar mass is 46. So if you do a little bit of a calculation, a 50 ml measure of vodka, which I understand is a fairly small measure these days, contains about 15.6 grams of ethanol. Okay, so we'll remember that number, 15.6 grams. If we work that out in moles using the simple calculation, it works out about 0.34 moles of ethanol. Now this is NAD. It's quite a big molecule. It's got a molar mass of 663. So bear in mind that 0.34 moles of alcohol we talked about early on is done twice. So for each time 0.34 moles of alcohol is metabolized by this route, it converts 449.7 grams of NAD from NADH for each shot of vodka. So 2 NAD converted to 2 NADH, which means we've converted 449.7 grams of NAD into a similar amount of NADH. There's not a difference between the two molecules, so we can say the weight's about the same. So that's a lot. Whoops, where are we? Yep, there we are. So 449.7 grams of NAD used up for each 50 ml shot of vodka. The liver only weighs about 1.45 kilograms, less in women who have a smaller liver, both in terms of weight and proportionally as well for their body size. So that's quite a lot. The slide from here is from the Pill for Nutrition lecture, which you may remember. As discussed before, fat has the highest energy because being very hydrophobic, it is essentially anhydrous. Proteins and carbohydrates are always found associated with large amounts of water, often bound by hydrogen bonding. Ethanol is infinitely soluble in water. But the problem is we have no biochemical mechanism for controlling the metabolism of alcohol, when our system is preferentially used for energy metabolism, so we don't use either recently consumed or stored lipids and carbohydrates, which may of course have long-term physiological consequences, specifically weight gain and other issues as well. There's the little slide we needed. Methanol is a food safety issue. It follows a similar reaction path, but this case started with methanol. Methanol is found in counterfeit alcohols such as that shown. When metabolized, it produces the highly toxic formaldehyde and also formic acid. And consumption of methanol can lead to serious issues, including blindness and even death due to these toxic metabolic processes. Okay, so there we are. That completes this series. Thanks for listening.